Hey everyone, it's Mr. Sinti, and I want to talk to you today about a really cool technique that we can use in the lab to separate proteins from one another. It's called chromatography, and in particular it's called GFP chromatography because we're trying to separate in the experiment the green fluorescent protein. But in fact we're going to be using a general type of chromatography that can be used to separate a particular type of protein, and it's called hydrophobic interaction chromatography, or HIC. And so let's just jump right into this interesting discussion. Let's go there, and then let's go there. And so let me overview the protocol, although I'll go into the details in a moment. If we are successful at transforming E. coli with our P-glow plasmid, the colonies should appear as green little colonies in our petri dish, especially when we've induced them with arabinose sugar. What we're going to do is then take those colonies and grow them up a little bit in a liquid media, and then overnight to make sure that the numbers are really high and they're expressing lots of protein. And then what we're going to do is in the next day is put them in a microcentrifuge and spin them down and pellet the cells. I don't know if you can see this really clearly, but the cells, let me sort of try to emphasize it, the cells over here in this test tube, in the microcentrifuge tube, are going to be made up of this supernatant, which is a lot of the LB broth, and the cells will be pelleted on the bottom of the microcentrifuge tube as this green blob. And in fact, there's a lot of E. coli cells and they're producing a lot of green fluorescent protein. You're like, well, what's the point of this whole thing? The point is the bacteria produce a lot of proteins. They're not just producing the green fluorescent protein that we engineered into them from a previous experiment. They're producing thousands of endogenous proteins that they normally produce. And so it's up to us to somehow get the green fluorescent protein out of these cells. And so you might have guessed it, what we have to do is crack the cells open. So we need to apply an enzyme, a lysozyme enzyme that frees them up. And so first we have to remove the supernatant. So you'd want to remove the supernatant from this. And then what remains is what's called the bacterial lysate. And so it's all the protein from inside the bacteria. And then what we're going to do is, better pictures coming, we're going to actually put them through a column chromatography using hydrophobic beads or resins, and we're going to separate the proteins from one another, and hopefully the final test tube is going to have our purified green fluorescent protein, which is actually green, not blue. And so here's our dilemma. This is you, so to speak, and you have your hand lens and you're looking through all these proteins that the bacteria normally produce. And so we're looking for just one. It's sort of like finding a needle in a haystack, so to speak. So they produce thousands of protein on their own. So what we're going to do is be using these columns, and they're these plastic tubes. And what this is a glass test tube. And we're going to put this column in there. And what this sort of gray area is are these little tiny beads. And what's fascinating is that in biotechnology we can use different kinds of columns depending on the need. Like for example, the two main kinds of, pardon me for this, this is my favorite thing that happens when I'm making a podcast. The two main types of chromatography are something called size exclusion, and I'm not going to go into that because that's not the kind of chromatography that we're doing. But basically, I will for a second. Size exclusion would be using a type of beads that reminds me of like wiffle balls. And depending on what we're trying to isolate, if the molecules are very small, they'll go inside the beads, or if they're large, they'll go past the beads. And we can use that size to separate small from large. But in this case, we're using something called hydrophobic interaction. And so let's just cut right to the chase. We know that the green fluorescent protein is extremely hydrophobic, and we can use that. And so when we add our bacterial lysate to this, and so here, what does that mean? That means our bacteria that have been cracked open that are producing the green fluorescent protein, so this juice. So when we add that, the green fluorescent proteins are going to cling to the sides of these beads because it's also hydrophobic, especially in a high salt buffer. And then we're going to add low salt buffers sequentially and remove all the other proteins that the bacteria produce, and then ultimately we're going to try to release the green fluorescent protein or elute it and then capture it and purify it. And so let's take a look at that process. So ultimately this is our goal, to put in a microcentrifuge tube purified green fluorescent protein. Not that that green fluorescent protein is of significance, because it really isn't. It just proves a point. It's kind of like this experiment is showing what is the potential of if we can add a foreign gene into a bacteria and it produces a lot of protein, and then we can purify it. We can use this protein for medicinal purposes. Like, for example, if this was insulin, we can then give this to patients with diabetes. And so it's a really powerful, very important technique to be able to isolate proteins from bacterial proteins. Very, very important. So this particular type of chromatography, again, it's HIC, so hydrophobic. And so first thing that we want to do is add the bacteria juice that are producing the green fluorescent protein to the column, which is that plastic tube matrix, because it has the beads in it. And we're going to use a very, very, very high salt buffer. And then what we're going to do is add a little bit of a less hydrophobic, less salt. And what that's going to do is going to remove any of the less hydrophobic proteins from the column. And then we're going to add a no salt at all. And that's going to pull the green fluorescent protein from the column. And so it's kind of a sequential thing. We're going from high to low to no salt. So that's what those solutions are going to be. And so I like this picture a lot. You could see over here how the green fluorescent protein is clinging to the beads, and then all the other endogenous proteins are clinging at first in the high salt. But then when you begin to wash them with the low salt, what's going to happen is that the endogenous proteins will be released from the column, and our green fluorescent protein should cling, hopefully. And then you're like, okay, that's great. So what we need to do is actually have 3 test tubes. We need to have one that releases the storage buffer, and then number 2 is we collect all of these proteins that we don't really want, and then the third one is where we actually remove the green fluorescent protein. So we're going to need 3 tubes. What's interesting is if this thing's glowing, you should be able to see this with an ultraviolet light in the column, and so hopefully we'll be able to check that out, and I have a little video to show you. And then finally, the TE buffer, which contains no salt at all, is added, the elution buffer, if you will, and then that will allow green fluorescent protein to flow through the column, and it goes into our microcentrifuge tube, and then it's kind of cool. This is a picture of the green fluorescent protein, and it's kind of like these beta sheets in it, and it's sort of cylindrical, and ultraviolet light comes inside and it sort of fluoresces. It's really neat. And so let's take it over to here. Let me show you this. This is kind of a cool rendition of what we're going to do. Here's the column in real life, and so to prepare the column you have to remove the little stopper on the bottom and take the cap off. And so you might notice that we're going to put it in a test tube. This is a little piece of paper that we're going to try to wedge in to make sure that it doesn't form an airtight seal, because then none of the juice will flow out. And so it takes a little while. Do you see this is the matrix down here of beads, and then on the bottom it's going to drip, 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 drip like this, kind of slowly. And so what this is the storage buffer. And so we're going to then take our bacteria lysate. So this is bacteria that's making green fluorescent protein, and we're going to put it in there. And we're going to add it in very slowly right above the matrix. We don't want to disturb the beads. And so we're adding, so this is not really bacteria, but basically the proteins of the bacteria. And when you put an ultraviolet light behind it, you can literally see the green. Do you see this? The green fluorescent protein is literally clinging to the top beads. And so now we're like, okay, What about all the other bacteria protein? Like, we've got to get rid of those. And so now we're going to add a low salt, the column wash, we're going to add a low salt solution to this. And so as that is added, and if you're doing this in a dark room with ultraviolet light, you can actually see how the green fluorescent protein will remain clinging and the other proteins will drip out. And then finally you add the no salt buffer. And when you add no salt buffer to the column, that's when you're going to get the green fluorescent protein to elute through the resin. And so it's really cool. You can actually see the protein like moving down, moving down, moving down. It's very exciting. So then you're going to want to capture it. And so see, there it is dripping out. See how proud you are. It's sort of like a baby being delivered or something. Maybe not that extreme. Here's a green fluorescent protein that's dripping out, and then you're going to want to collect it in a microcentrifuge tube because it might be more convenient to store it this way. And so there you have it, the purification of the green fluorescent protein using hydrophobic interaction chromatography. I hope you enjoyed watching that, and I hope it was helpful. Thanks.